name is Gosia. I come from Krakow in Poland, where I studied biochemistry at the Jagiellonian University, and in the past I did Erasmus Plus internship in Leuven, Belgium and Utrecht, the Netherlands. My main background is immunology with a focus on host-pathogen interactions.
So hello, I'm Elena. I come from Genoa, where I took my bachelor in life sciences, and then I decided to move to Pavia, another Italian city, to take a more specific master in molecular biology and genetics.
Hi, my name is Astrid. In the Netherlands, where I also obtained my bachelor's degree in biomedical sciences and a master's degree in infection and immunity.
Hi, I am Rita. I am Portuguese. I did my studies in Portugal in the University of Coimbra, where I graduated from biochemistry, and I did my master's degree in pharmaceutical biotechnology.
I did my internship in the UMC in Utrecht, and I found out about this project between UMC and GSK Vaccine in Siena, and I found it's pretty interesting since it's not so common to have a PhD between pharma and academia.
Okay, so during my last year of university, I heard about the GSK site in Siena, and I decided that I wanted to experience some scientific research out of the academia and in a center of excellence. So I managed to obtain an internship, and during this time I learned about the DISSection grant on the Euraccess portal. And at that time I wasn't really sure whether I wanted to do a doctorate degree and where, so this felt really like the perfect opportunity for me to explore both the academia and the industry, my country and a foreign one, and thus challenge myself and collect more information in general about the EU scientific panorama.
I was finishing my master's in Utrecht when I heard about this opportunity. I was excited by the opportunity to experience working in both industry and academia. And next to that, I was also intrigued by the challenge to conduct research in 2 different labs across 2 different countries during my PhD.
I wanted to work in a company, or at least experience the work in a company. So I thought I could look for positions for a PhD abroad and in a company. A lot of requirements, so I didn't really find a lot of positions. But after some search, I found a couple of them and I applied to them. And I was really happy I was accepted to work under this grant.
My project is focused on the interaction between skin-resident immune cells and Staphylococcus aureus. So during the first part of my PhD in Utrecht, I look at the interaction between antigen-presenting cells, especially lectin receptors, and carbohydrates on the surface of bacterium. And I have identified one of the interaction between MGL receptor and the sugars on Staphylococcus aureus. Whereas now I work on the T cells responses, and I continue on the project that was started by Astrid.
So my main subject in the project is the phagocytosis of Staphylococcus aureus by professional phagocytes. And despite approaching the topics in a very different way while I was in the UMC Utrecht or in GSK, I can definitely say that I acquired a large scientific background about the topic and a lot of practical skills to tackle scientific questions about it.
My project is focused on the interaction between the immune system with Staphylococcus aureus, specifically in the skin. So during the first half of my PhD in GSK, I studied T-cell responses to Staph aureus, zooming in on a specialized memory T-cell subset that resides in human skin. I continued my research in Utrecht, where I studied the immune recognition of Staph aureus by antibodies and Langerhans cells, which are key antigen-presenting cells in the skin.
So I started my PhD in GSK vaccines in Siena, and there I was interested on studying how Staphylococcus aureus colonizes and infects human skin. More specifically, what I did was to study gene expression of the most important virulence factors of this bacterium and try to pinpoint which factors were more expressed by S. aureus within the infected tissue. Currently, I am working at the UNC in Utrecht, and my work here is a little bit different. So, as I am interested on virulence factors of SRS, we thought we could zoom in and focus our studies in one of the most important virulence factors of S. aureus, and this is staphylococcal protein A. So protein A is an immune evasion factor. So it means that protein A helps the bacteria to hide from our defenses. For that, protein A binds to antibodies in the wrong orientation, and therefore it prevents immune cells to recognize the invading bacteria. So now I am investigating whether this binding to antibodies in an incorrect orientation, if it also leads to less complement activation, which functions as an immune defense mechanism.
I am happy to work on a scientifically relevant model of the skin infection that could potentially give some insights into the skin immunity, and especially the fact that I am able to do it in 2 different institutions. It's quite unique. So during these 3 years of my PhD, I have already published 3 papers.
My key results elucidate the importance of antibodies and complement in the phagocytosis of S. aureus, which is thought to be a really important and pivotal mechanism of pathogen clearance by the host.
So I think gaining insights into the pathogenesis of Staph aureus skin infections is very important. Since it's still the leading cause of skin and soft tissue infections.
Information about the pathogenicity of the bacteria in the context of the infection is very important for us to understand how infection occurs. But it's also very important, for example, when you want to select proteins for a vaccine formulation, because you want to confirm that the bacteria effectively expresses this protein in the infected tissue. Understanding the mechanism by which protein A evades our immune defenses, it will help us to afterwards develop therapies to prevent this effect.
So it was the first time for me to work in the pharmaceutical company, so this type of work differs a lot from the academia, and you need to really change your mindset, but simultaneously you gain a lot of experience both scientifically but also a lot of soft skills. And another important thing is the collaboration, because we are PhD students that need to collaborate with each other in order to publish. We have to collaborate with our supervisors, so sometimes it's a little bit hard, but when it works, it gives us a lot of satisfaction.
I feel like I definitely acquired all the lab skills that I need in the field of microbiology and immunology, especially flow cytometry and microscopy, to perform all my phagocytosis assays and tackle also other scientific questions. I feel like I'm becoming an independent scientist, so I can formulate hypotheses, I can have opinions, and I feel like I can definitely acquire new knowledge very quickly compared to before. So furthermore, this PhD gave me the unique opportunity to experience 2 centers of excellence in the academic or in the industrial sector, and from the privileged point of view of a student. So in the academic experience, it represented a really steep learning curve in the scientific topic that I work on, while in the industry it was different, and I'm learning to be more result-oriented and goal-oriented and limited by strict international regulations.
So this project has provided me with a great platform for personal and professional development. We could experience working in a pharmaceutical company, follow courses, expand our network, and also attend international conferences. It was quite challenging to move during our PhD, but this experience did help me improve on key skills such as time management, but also adaptability and perseverance.
I learned a lot of techniques since I started my PhD. I had my first experience working with tissues when I worked with human skin explants in GSK. And while in UMC, I gained experience on performing more in vitro assays as complement activation and phagocytosis assays. So the work in these two groups with so different expertise, it really enriched my lab experience. Additionally, during these 3 years, I met and worked with many people, and therefore this project was also a good platform for me to expand my professional network. And I feel very lucky. I had the opportunity to experience both academic and industrial environments in 2 different countries during my PhD. But I have to say, it was challenging to have to move and also challenging to coordinate my project with the project of the other fellows of this grant. Eventually, this project really contributes for me to improve my time management skills and also to improve other soft skills.
I personally love traveling, so doing a PhD in 2 foreign countries was a reason for me to apply for it. And it's obviously not easy to live apart from your family and friends, but during my PhD I met many great people that hopefully will stay my friends for a lifetime. And another important aspect is learning new culture of new countries, so both Italy and the Netherlands, and I'm happy to explore cuisine of both countries. So it is an experience that you cannot gain as a tourist.
So this project gave me the tremendous opportunity to understand better my necessities, both in terms of lifestyle and work preferences, while receiving high quality training in the meantime. So sometimes it was very hard to change culture and work environment very, very quickly, but I think that this is a valuable lesson for the future and made me grow up professionally a lot.
So moving to a different country really teaches you a lot about yourself. You have to cope with cultural changes, you're meeting new people, you're learning a new language. And the biggest lesson I learned from living in Italy as a Dutch person is that it's okay to take it easy sometimes and just enjoy life as it is at the moment, so with good food and good company.
It was a great personal experience. I'm really happy I accepted the challenge to live and work in 2 so different countries. I fell in love with Italian cuisine and the region of Tuscany, but also with the tulip fields in the Netherlands and all the windmills. As the culture of the 2 countries is so different, I think I'm more open-minded after this experience. And I also learned a little bit of Italian and Dutch, and I made good friends that I hope to continue in touch with.
In the future, I definitely see myself in the scientific field. However, I don't know for now whether I would choose pharma or academia, but thanks to DISSection, I have insights in both environments and it's easier to make a choice. ITNs are pretty demanding projects, and especially from the organizational point of view, but I think it's worth to apply for because they give you a lot of training and high quality research, so at the end you get a lot of irreplaceable experience.
So in the future, I see myself working in a pharma or healthcare related industry. And there are also many different and unconventional positions that I would like to explore apart from the classical laboratory scientist. And my take-home message for students who wish to apply to an EAD project is to stay open for the opportunities, be brave during difficult times, and enjoy the pathway.
I can continue my career in science in one way or another. And my message for future ITN students would be to try to see this experience as a great learning opportunity. So try to learn as much as you can, both professionally and personally, and most importantly also enjoy the experience.
I would like to continue my career in science. I really enjoy laboratory work and also the challenge of interpretation and unexpected results and try to find an explanation for it. I still didn't decide whether I would prefer to work in academia or industry. Something I decided is that I would like to go back to Portugal, at least for the coming years, and work there. So about my take-home message for the students that want to apply to an ITN project is to learn as much as you can from academia and industrial environments, but also from the cultural aspects of the countries you are living in. And of course, enjoy the journey.